Imagine a tiny, resilient organism clinging to a rocky outcrop, enduring the harshest of climates from the scorching desert to the icy Arctic. This is a lichen, a remarkable partnership between a fungus and a photosynthetic partner, typically algae or cyanobacteria. These humble organisms are nature's pioneers, thriving in places where few others can, and serving as living indicators of environmental health. Lichens come in an astonishing variety of forms, from crusty patches on stones to delicate leafy structures hanging from trees. They are silent witnesses to the passage of time, some living for thousands of years, and have intrigued scientists for their unique ability to weather extreme conditions, even surviving the vacuum of space. Their story is a testament to the power of collaboration in nature, where different organisms join forces to create a life form that is greater than the sum of its parts. Step into the world of lichens, where fungi and algae merge into nature's masterful survivalists, creating intricate mosaics that colonize the harshest terrains and withstand the test of time, illustrating the extraordinary beauty and resilience born from collaboration. Lichens are among the most captivating examples of symbiosis in nature, arising from a partnership between two distinct organisms that, together, form a new entity capable of enduring conditions neither could withstand alone. At the heart of every lichen is a complex relationship between a fungus, known as the mycobiont, and a photosynthetic partner, the photobiont. The mycobiont provides the structural framework and protective environment, while the photobiont, usually a green alga or cyanobacterium, harnesses the sun's energy to produce food. This extraordinary collaboration not only allows lichens to thrive on bare rocks, tree bark, and even human-made surfaces, but also creates intricate and varied forms that contribute significantly to their ecological niches. Understanding the distinct roles and interactions of these components is key to appreciating how lichens successfully inhabit some of the planet's most challenging environments. Each layer of the lichen thallus plays a unique role in supporting the organism's survival, from protection to nutrition. The cortex, which is the protective outer layer, consists of two parts. The upper cortex, which is the topmost layer of the lichen, directly exposed to the environment. It acts as a protective barrier against environmental stressors, such as UV radiation, desiccation, and mechanical damage. It often contains pigments and secondary metabolites that protect against intense sunlight and herbivory. And the lower cortex, which is the bottom layer, if present, facing the substrate like rock or tree bark, that provides additional protection and can anchor the lichen to its substrate using rhizines, which are root-like structures in some species. In crustose lichens, the lower cortex may be absent, with the medulla directly attached to the substrate. The photobiont layer in lichens is a crucial zone located just beneath the upper cortex. This layer contains the photosynthetic partner, which can be green algae or cyanobacteria. These photobiont cells convert sunlight, water, and carbon dioxide into carbohydrates through photosynthesis, providing essential nutrients that sustain both partners in the lichen symbiosis. The fungal hyphae surrounding the photobiont cells help protect them and facilitate the transfer of nutrients throughout the lichen's body. This interaction is vital for the lichen's survival and ability to colonize various environments. The third layer is the medulla, which is the supportive and functional core. It lies between the cortex and the photobiont layer, or beneath the photobiont layer in some lichens. It consists of loosely woven fungal hyphae and acts as a storage area for water and nutrients and facilitates gas exchange. Its spongy texture helps the lichen absorb and retain moisture, aiding in the regulation of water balance and respiration. Lichens exist in one of the mentioned growth forms. Crustose grow across the substrate. Foliose are flat, leaf-like sheets of tissues and not bound closely. Squamulose are closely clustered and lit flattened pebble units. Fruticose are freely available in standing branching tubes.